we're going to shift gears now into a new theme, which is around biosafety, security, and ethics. And we've got a couple of wonderful speakers now that Manu has helped us to frame wonderful tools for access. Now we have to think about how do we ultimately engage ethically with our communities, how do we do it safely, and how do we do it securely. So it is a pleasure to introduce our next speakers. We have joining us Todd Kuiken and Dan Grushkin, who have been working for a very long time on a wonderful handbook around biosafety, which they're going to tell us all about. So everybody, please join me. We're going to try the unmute thing again. Join me in welcoming Todd Kuiken and Dan Grushkin, everybody. Thank you, guys. Yes. All right, go for it, guys.
So thanks for having us. Dan, I just want to make sure that you're there. Dan's going to take off the second sort of portion of this. But I need to start with my public service announcement, particular people in the US, and just to remind you that Devo says to vote, and that the deadlines for registering to vote in the US are all coming up rapidly. So make sure you're all registered, because most of the things we're talking about today will get impacted by who our leaders are. So it's really important to register and to vote. So with that, let me move us into this. Let me try to share my screen here. I think I can do that, right, David?
You can. You are a co-host. You have the power.
Awesome. That power feels great. So what we thought we'd do is to talk about the handbook that's taken that we've been working on, and a number of the people in the community have been working on. Some of you have had visits from us over the last couple of years to get input from this. And what I wanted to do was—
Todd, I'm sorry, do you want to just go into present slide mode? We're seeing your
yes.
speaker view.
Is that better?
Perfect.
Yeah, that works, Todd.
Okay. So, but I wanted to talk about that this idea of responsibility and respect for the science has really been a part of this community from the very beginning. And we thought we'd kind of bring you through the history a bit of where we've been and where we're going. And so I just wanted to throw up these pictures, which we think may be the first pictures of the first official DIYbio meetup in the US, which happened in 2008. I'm not sure if anyone who's here today was actually in those pictures, but if you are, maybe identify yourself in the chat box. But similar events like this, as you know, were occurring across the globe. And as David mentioned yesterday, around 2010, at least in the US, some of the first community labs began to open. And I just wanted to give a shout out to Jason Bobe and Mackenzie Cowell here, who were the co-founders of what was maybe one of the first sort of meetup portals on the internet, which was DIYbio.org. So I just want to make sure we remember our past as we keep moving forward. So this is a picture of the first code of conduct meeting that we held back in 2011 in Europe. And this was sponsored by the Alfred P. Sloan Foundation. And I want to give them a shout out as well. They were one of the first foundations that recognized the power and importance of this community and provided a lot of resources over the years to help us do a lot of this work. But I just want you to sort of take stock of these pictures. This is pretty much how big the community was in Europe. These were the heads of the labs and the groups. And compare that just to the amount of video screens that you see on your screen right now and how much this community has grown over the years. And what we did here was similar to what Alex Perlman has been running for the past couple of years at the Bio Summit, which was to gather our thoughts around how we wanted to operate around safety, security, and ethics. And we repeated these exercises in the US as well. And as many of you have known and seen, we developed these codes of conduct back in 2011, which were great at the time and I think have been great to sort of help evolve the community as we've grown. And that grew from those conversations into something we had, which was the Ask a Biosafety Officer web portal, which enabled people to ask biosafety questions. And this was backed up by the American Biological Safety Officers Association, who are expert biosafety officers, who also recognized the power and potential of this community and offered up their expertise to make sure that we were all able to sort of run our experiments safely and responsibly. And that has grown into something we ran last summer, which was the Biosafety Bootcamp, which some of you were at, which again, ABSA developed a special course for us to train ourselves and then be able to train other members in our community around how to actually run our laboratories in a safe and responsible manner, and in some instances, make sure that we're following some of the local regulations and guidelines. And here's a snapshot of all these beautiful folks from 15 labs from across the globe that participated in this. And we're hoping to be able to continue to do this further. But these types of exercises also require some resources. So I want to shout out the Open Philanthropy Project who helped fund this project. And here's just some pictures of some of the folks you probably recognize sort of huddled around a biosafety cabinet. And more recently, which many of you I think are participating, is the Open COVID-19 Initiative. And this I think is a really great example to show the progression of our community. And one of the first things that started up within this project was a biosafety security committee that was able to start to answer the questions as they came up. And a new study that's happening that Alex Perlman and myself and so many others are a part of is to sort of study how this actually works in this new format of Joggle in terms of this very widely distributed types of projects, and how do we make sure that we have the proper sort of ethics and safety standards in place that our community, which has some unique circumstances to it, can continue to operate. So that leads us up to the handbook, which I'm now going to turn over to Dan.
Thanks, Todd. That was a quick summary of 12 years of work. So one of the, I think, culminating moments in all of this work around ethics and biosafety and security is something new that we're introducing to the community today. And I think this is a really, really important resource that we've developed with lab heads and experts in the biosafety community from across the world. So we've created over the last 3 years something called the Community Biology Biosafety Handbook. Now this is a 255-page document that's right now sitting in a beta form in a Google Doc. But why don't we go to the next slide? It contains I think all of the expertise and knowledge that we as folks who have been running labs over the last decade have compiled. Everything ranging from, and let's go to the next slide, everything ranging from how to put on your gloves properly and sanitarily to, next slide, how to do an outreach event that's safe and also effective, to really everything in between. It covers everything from shipping chemicals, collecting chemicals, getting new equipment, how to interview a new lab member, really just the entire gamut. Let's go to the next slide. I think we really need to thank the authors of this. This was really a long journey with these folks. So the authors are up top and then the editors are on the bottom. Now these are folks who are both experts in lab management and just folks who are leaders in this community and have been for years, who have really put together their expertise and knowledge. And then us folks on the bottom include also David Gillum, who is a biosafety officer and a leader within the American Biosafety Association. So the document has also been vetted by him as well. The thing that we're doing with this document, we've done as much as we can. It now lives in a Google Doc, but it's time for us to move it from our hands into the community. And so let's do the next slide. This is, by the way, this is us working on it. This was like the final push, 3 days of basically Todd locking us in a room, going through every single line of it. And we can go to the next slide. So we're now at the point where we're going to be handing this over to the community. And the community really needs to decide, how do they want to use this as a resource? The way I see it is that this is really a baseline document for how to safely run either at your own lab at home wherever it might be, or even a community biology lab. It contains all of that material. But of course, it's just really the first step. So we only know so much of the regulations that are out there. The technology that we've been addressing is the technology that is a snapshot of now. But for this thing to really live and thrive and be a resource to the community, it needs to go into the community's hands and then have the community update it, add their regional regulations, add, you know, our group response to a new technology, how we want to address it safely, and then carry it forward. So my hope is that, yes, we have 255 pages. My hope is that if we come back to this in 5 years, there'll be far more pages that are very specific to individual labs, but also really something that anyone who's starting can use and essentially build their lab based on these protocols. That's my fingers crossed hope. Later this afternoon at 2 p.m. Eastern, we're going to be coming together as a group to look at it, review this document just to see what's in it, and then start to think really deeply about how we're going to do governance around this document. Obviously, we don't want to give people bad advice in this. We want it to be a useful resource. And so there needs to be a way for us as a community to basically edit and caretake this document and make sure that it gets out there and is a resource to all. So right now it's sitting in a portal on GenSpace's website, which you can see on the screen, but really any lab, any individual could link to it on their own website, on their own page, on their own social media, however they want to, but it really needs to get out there. So we're really excited about it. I think this is really a hallmark after 12 years of work for Todd and me, and certainly the authors. And I'd like to think of it as a hallmark for the community as a space of really responsible and thoughtful science. That's my 2 cents.
Wonderful. Gentlemen, that was really, really fantastic. And so a reminder for everybody, we're going to have a much longer 90-minute block on this. So please join us. Actually, the chat, there are some really fantastic ideas already going in there about this. So please continue this conversation with Dan and with Todd later on this afternoon. So can we get some snaps and some applause and some emoji love for Todd and Dan, not only for this presentation, but just for your guys' leadership over all of these years and the tremendous work on this handbook. Thank you guys so much. And please go to the lysosome at 2 o'clock to continue that conversation. Thanks, guys.
Thanks, everybody.
Okay, and up next, and thank you also for being exactly on time as well. Up next, we have Sarah Ware from the BioBlaze Community Lab. And Sarah's going to be talking about responsible ways for us to engage in disclosure and peer review. And Sarah, again, has been working really, really hard with a number of wonderful folks and leaders in our community on this disclosure work. So Sarah, I will turn it over to you.
Hi, thank you. I'm excited to be here, although virtually. I mean, I know everyone misses being in person, and me included. It's one of my favorite times of the year to go to BioSummit, but thankfully we can meet in this virtual format and in my heart. So I'm really happy to be here and talking to you guys. This is my 4th BioSummit. So I'm really looking forward to Todd and Dan's session that's coming up at 2 p.m. And so I hope you guys are going to be there to talk about the Biosafety Handbook and how we can more make it our own, although I'm sure they've done an amazing job of that on our behalf so far. Well, so we have collectively created that Biosafety Handbook up to this point. I mean, Todd visited BioBlaze, and I know he's visited some of your community labs, collecting information and trying to make that work for our community. And I'd also like for you to consider coming directly after the session this afternoon on the Biosafety Handbook to another session that I'm involved in with Amanda Plimpton and Tessa Alexanian. And that is a very similar topic, but we also need your input to make it collective and make it work for our community. And so that session is at 3:45 and this afternoon, and it's on cultivating a culture of responsible disclosure and peer review. So there are three closely related topics that we'll be encompassing, including Todd and Dan. And those topics are, of course, biosafety, talking about the Biosafety Handbook, but also biosecurity. And that's what Tessa is going to be talking about with responsible disclosure in our community. And the third very closely related topic is bioethics. And so Amanda Plimpton will be leading that portion of our breakout session. We'll be talking about a peer review board toward the formation of an IRB, or an institutional review board. So the theme of BioSummit this year is governance, and we as a biohacking community, we're getting organized, and it's really kind of interesting, and it's kind of hard for me to wrap my mind around that, how that's possible, because all of us are so independent and have our own way of thinking and doing things, but somehow we are collectively becoming organized, and we're making it work relatively well. So anyway, we have our own conferences, of course. We have a Biosafety Handbook now. We have codes of ethics. And so these efforts are not, you know, we're not doing these things and we're not getting organized in order to stifle your ideas and your research efforts and your innovation. Rather, we're doing these things to support your ideas and to support your research and help move it forward. We all want to be safe. We all want to be mindful of biosecurity issues. We all want to be doing ethical research. So we have kind of come up with a name called CLEAR. Very recently, there's a group of us, I guess it's mostly like Todd, and then Alex Perlman and I have been kind of kicking around this idea of a peer review board or an institutional review board for the biohacking community for about maybe a year and a half. And we're gaining a lot of momentum in that, and so that's great. But we have this name now called CLEAR. And so it stands for Community-Led Ethics Research and Assessment. And we hope to help you guys bridge the gap. It's actually a ravine of getting your great ideas to the masses. And right now, there's really not a forum to do that. Right now, I mean, I'm just going to say it clearly, right now, I mean, if you have a great idea in human health, and you want to get it to the masses, you're going to hit a wall, and that's just the reality of it for the moment, because we don't have an institutional review board. We don't have this first piece of information that we need to have approval of before going any further. So we want you guys to, you know, as Dan had said, like, we're handing this over to the community. We've done a little bit with it. We're handing it over to you guys, and we hope that you will join us at 3:45, that you will weigh in on, you know, what do you think you could get out of such a peer review board? Who do you think needs to sit on that review board? What would prevent you from submitting a project to this review board and getting some input? Yeah, so that's about it. I hope to see you guys there at 3:45, and also, of course, go to Todd and Dan's session at 2. Thanks, guys.
Sarah, thank you so much for sharing, and also for all of your leadership. Really, this is amazing. And so join, again, this is all going to be happening. Is it in the Lysosome, guys? I think it is. Yeah. It'll be in the Lysosome starting at 2 o'clock. There's going to be fantastic sessions all on these themes and topics. So please join. I think just following up on my news observation around really transferring ownership to communities, I think this is, you know, we have innovation responsibilities, but also, of course, safety, ethics and security responsibilities as well. So thank you guys so much. And thank you, Sarah, again, for sharing. Can we get a round of applause and some snaps, some emoji love for Sarah? Thank you guys. Really appreciate it. Okay, and let's transition into our final talk for the session this morning before our next break. So it is my privilege to welcome my colleague at the Media Lab, Professor Kevin Esvelt. So I feel like, much like with Manu, this particular talk is also maybe 4 years in the making. Kevin also has had an extremely busy travel schedule, much like Manu. And so again, Kevin, I was telling Manu this morning, in one way, the pandemic has also, obviously created massive problems for society, but in a sliver of a silver lining, it has also made it easier for you actually to join BioSummit this year. So it's so awesome to have you. The other really funny thing too is we were kind of working on the title for Kevin's talk, and I'd originally put up Global Catastrophic Risk was my original title. And then Kevin messaged me yesterday. He's like, you should put mitigating in front of that. So it should be mitigating global catastrophic risks, which I thought was a very important addition to your talk title. So with that, Kevin, it's a real honor to have you here, and just really thrilled to hear about both your thoughts and your assessment of what are some of these risks that as a community we should be thinking about. But again, I think as we were talking about, I think last year, I'm really getting excited about having this community take on a leadership role and being able to assume some of those responsibilities, as you've been hearing from our speakers earlier this morning. So everybody, can you join me in giving some claps and some snaps and some emoji love for Kevin Esvelt? So thank you so much, Kevin. Take it away.
All right, thank you, David. And hello, everyone. So I'm going to give a bit of a perspective on autonomous biology. Now, obviously, all biology is autonomous, but as is evidenced by the fact that we are, in fact, here indoors and not all together in one place, autonomous biology of the kind that spreads uncontrollably beyond human control has some degree of risks. And this is something that I was largely oblivious to until a few years back. And I really want to share a bit about how that changed for me and why I think it's so important that the community bio movement take a leading role in changing how our society looks at this kind of biotechnology. So I actually added a community mitigation of catastrophic biorisks there.
Love it.
And, okay. So for context, 8 or so years ago, no human in the entire world had ever even imagined that a single researcher could plausibly edit the genomes of an entire species. The very idea was completely absent from science fiction. If you can find a counterexample, please let me know. To date, no one has. And I had played a very minor role in developing CRISPR, and I was wondering, will we ever edit wild species? And I thought, well, what would happen if instead of just inserting stuff in the genome that we want, what if we also encoded the CRISPR genome editing system? So the cells of an organism in the germline could do CRISPR genome editing on their own. If they did, then when an edited organism mated with a wild type organism, all the offspring would inherit. And in the germline cells of the offspring, CRISPR system would copy the change, overwriting the original wild version, thereby ensuring the next generation inherits, and the next generation, and the next, and the next. So this is what we now call CRISPR-based gene drive. And gene drive is, of course, a naturally occurring phenomenon. There's broken gene drive remnants all over our genomes and in the genomes of just about every other species on the planet. But with CRISPR, we could plausibly do this deliberately. And this could be done for some amazing things that's raised a lot of challenges involving how do we go about relating to communities when our technology isn't just something that's going to be put into a product, like a biomedical product that people will be able to get recommended by their doctor, and then they can say yes or no. If your doctor recommends something that we develop, you can decline. But if we develop a technology that will change the shared environment, that's very different. Even if your community votes on it and you vote against it, you're going to be affected no matter what. So we should give you a voice in how we develop that technology, just like people should have a voice in discussions of what the laws should be, because it's the same kind of effect. But that's not the focus I want to talk about today. Rather, it's that a single researcher can build one of these things. So if any of you have worked in fruit flies and can do microinjection in fruit fly eggs, you could just build one of these things and you could let it go. And according to our models, it takes very, very few organisms introduced in order for it to take off and spread. And this is an extremely weak gene drive system that only copies itself half the time and otherwise makes resistant alleles. It never gets above 30 to 40% frequency in a population. But even so, vanishingly few organisms are required to be released into a population for it to just take off and spread. And this is a problem because it's something that one person could do that could really impact just about everyone. There is what's called the unilateralist curse. And the idea is if any one person can do something that affects everybody else, then with respect to governance, which is the theme of this summit, the outcome will be determined by the most risk-tolerant individual with access. And that's what Nick Bostrom formulated when he laid out this principle a few years ago. But I think it's actually worse than that, because it's not necessarily about risk tolerance. People might not be aware that what they're doing actually does pose a risk. They could be unaware of negative consequences. So it's either the most risk tolerant or the least aware. And we need to be concerned about both. And part of the reason is that, well, we were concerned that many other researchers might independently develop CRISPR-based gene drive systems as a lab tool, because it offers obvious inheritance advantages, without really realizing that it might actually take off and spread out of the lab. Hang on, I seem to have lost my picture. Can everyone still hear me?
Yep, we can still hear you, Kevin.
Can you see anything?
We see a mouse hand.
Yeah, there's only this mouse, right? There's a mouse hand, and I'm not getting anything. Interesting. Downside of having to run VMware when you're a Linux fan. All right, so I will just wing it then without that. Let's see.
We appreciate it. Yeah, no worries, Kevin. If you don't have the video, you can also just speak.
Yeah, so I will speak extemporaneously because it looks like VMware has crashed on my computer, and it possibly has brought Zoom with it. Otherwise, I would just switch back and you could and actually talk to you directly, but that doesn't seem to be working either. Well, it is what it is. Oh, boom. Now I can see all of you. Hello, everyone.
There you go. Yeah, go for it.
All right. So without the slides then, we tried to warn people that this might be a problem. And all of our first papers are concerning and getting regulation and governance and the need for safeguards and to make sure that people just didn't build these things, because in typical fruit fly lab, flies just fly around in the corridors all the time. And we failed miserably, because not a year later, people published in the same journal that we had issued the warning, a paper describing how they had done this without realizing that it could spread in the wild. So what this shows is that we really have few opportunities to warn others, and there isn't really much of a mentality of caution in the sciences. Now, fortunately, CRISPR-based gene drive favors defense. From a security perspective, it's slow, it takes generations to spread through a species. Even fast reproducing species, that's quite a long time. It's obvious in that if you sequence the genome, you cannot miss it. You will always be able to pick up a CRISPR-based gene drive. And once you see it, you can build a counter, one that will overwrite the problematic sequences and will in fact immunize other organisms and prevent them from acquiring it. So anything that is slow, obvious, and easily blocked is not much of a security threat. Sadly, that is probably not true of all biotechnology. As evidenced by COVID-19, some autonomous biotechnologies almost certainly do not favor defense, as evidenced by our civilization's struggle to deal with it. Which raises the question, are we safe against people making pandemics? And what I am concerned about is that with experience of gene drive, we're probably going to see scientists who are not even aware of the risk, making it possible for others to build such things. We're not just talking about accidents in the lab, people building gain-of-function viruses that could be plausible pandemics getting out. I'm talking about sharing that information, sequencing all the zoonotic viruses that we're aware of, making predictive models that will be able to evaluate how close a given zoonotic virus is from jumping species and becoming a new pandemic. And then sharing all of that information, which would then amount to instructions for making such a thing. Because as Feynman said, what I cannot create, I do not understand. What are our barriers against that? Well, in principle, for anyone to build a pandemic from scratch, what they need to know, they need to have the relevant skills, as many people on this call do. They need to know what to make, which is what I'm afraid the scientific community is going to disseminate. And they need to be able to get the DNA. And right now, only 80% of DNA gets screened by commercial providers. And there's a number of companies developing desktop synthesizers, which may be convenient for many community labs, depending on where you are in the world. But they can't be screened with current technology at all. And as a result, we might be in trouble. And I don't just mean in trouble in the civilizational sense of what would a world look like where people could make SARS-3, SARS-4, SARS-5, SARS-6, SARS-7, SARS-8, all the way up. It's a lot harder to make and distribute a vaccine for a new pandemic virus than it is plausibly to make a new pandemic virus, if we are foolish enough to learn how to do that. But right now, the scientific community has a very individualist focused culture of free inquiry and dissemination of information regardless of consequences. And we ran headlong into this with CRISPR-based gene drive. The response was, okay, well, yes, we should use safeguards in the laboratory, but anything we discover should be disseminated regardless, and everyone should have access to this technology. Everyone should be able to make a CRISPR-based gene drive in any given species. That's what democratization of science means, right? Well, we don't necessarily want to democratize weapons of mass destruction, but that's what the current life sciences community in academia effectively advocates for without realizing that that's what their culture encourages. So this is why I think it is so important that all of this talks we've heard from Todd and Dan and Sarah, and what you're going to hear later on from Tessa and others, on how the community movement has been focused from the very beginning on safety and responsibility, and has been keenly aware of what public perception means for our ability to play with biology, to have fun, to learn, to discover, to explore. We can only do that if there is societal acceptance for that. And if some idiot or horrific evil person makes a pandemic, then the world will have no choice but to crack down on the ability to tinker with biology. Never mind the catastrophic consequences of civilization. And whereas academia is laden with all these incentives and a culture of this free inquiry and the assumption that all knowledge is worth having, community bio has been different from the beginning. And whenever journalists ask me, oh, well, what about the biohacking community? And I always say, I am far more worried about my colleagues in academia, because the community bio movement is actually aware that there are consequences to developing powerful new technologies. And they're aware that there are ethical considerations involved in tinkering with the stuff of life. And that is not something they teach you in a PhD program. NIH responsible conduct of research and all. So I would love to see it if the community bio movement took a leading role in not just continuing to pursue your movement towards doing the right thing in this regard, but so that you can be held up as an example and tell your friends and colleagues in academia that this is something we should be concerned about. Most scientists don't have friends who are thinking about these things. All of you are, and all of you have scientist friends. You could sway the broader scientific community on this issue and protect the right to freely explore with life in a safe and responsible manner that will promote human and animal flourishing into the future. That's my hope. And I would love to hear your thoughts on how we might make this possible. Please, given that I don't have my slides, so my email is esvelt@mit.edu. Please feel free to reach out to me with questions, suggestions, concerns. Just keep in mind, an information hazard is any information that, if disseminated, will cause harm. So if you're worried about some particular technology, don't just email me a description of it and say, hey, is this a global catastrophic risk? Because probably my email is being monitored. So be careful about, if you do stumble across that sort of thing, don't type it into an electronic device. You know, if you want to consult somebody about it, do so in a nice stroll at the appropriate 6-foot separation outdoors, well away from electronics. Because we don't know now that it's possible for people to build the equivalent of CRISPR-based gene drives on something like a pandemic virus. My hope is we will never acquire that knowledge, because I can't see how that would be good for us. We can defend against pandemics in other ways, and we should do that. But I think that there is a tremendous opportunity here for a cultural shift, because your culture is frankly far healthier than that of academia. Thanks.
Kevin, thank you so much for your remarks. And we've got a time for just a couple of questions and comments. I will say, Kevin, too, much as I was saying to Manu earlier, you should have, I think, gotten an invite into a Slack workspace. If not, I'll make sure. But we have one for the whole conference, and I think there's, whatever, 600 or so people on there. And it would be great to engage with you further there online asynchronously in the coming, weeks and however long. So let's definitely continue this conversation. And really, I think this combination of talks this morning with you culminating here, I think is really framed so powerfully the roles and responsibilities that we have as a community have to really embrace these topics. So there's been a number of really awesome questions in the chat. I just want to point out, I'll try to lift up a couple really quickly here. Alexandra earlier was asking about, how do we walk this fine line between free, accessible, open science, which is one of the core values, not only of our community, but I think of others that really, really love and engage in science, and some of these security risks that you're posing. And you've gotten to aspects of that, but I wonder if you can maybe provide a little bit more thought on that particular question of that fine line.
So this is a tremendously difficult one, because obviously it's hard to defend against something if you don't know what you're defending against, right? Sun Tzu. So but it's important to put the thread in larger context. Natural pandemics are a risk. There's roughly a 1% chance of something like COVID-19 every year, judging by history. And given that it can do this much damage, we do need to defend. So that can be, I'm personally super excited about environmental metagenomic sequencing. And this is something that the community bio movement could really lead in the citizen science perspective. Among other things, we're going to want to ensure that that monitoring is done well, which means spot checks, right? We want people to make synthetic DNA and dope it in the environment in some place, and then see if it shows up on the monitoring system. Make sure that it's being done correctly. But if we do something like that, we can always see it coming. And then once we see it, then we can plausibly defend against it. That's what I'm more worried about. But how do you walk this line? Take DNA synthesis. Suppose we want to screen all DNA synthesis, so you can't just plausibly make, make 1918 influenza, which I don't think would take off if you were to release it, but there's a small chance, and that is too much of a chance to take, right? Ideally, what we want to do is prevent people from ordering that unless they have permission from some kind of oversight body that is watching what they're doing and say, yeah, this is legitimate research. There's good reason for you to be doing that. In which case they should be able to get it with 0 delays. 0 delays. That's critically important. But supposing that we identify something that is dangerous, some way to make something that is nastier than a natural pandemic. Ideally, you want to screen against that, but we don't want to tell anyone what it is either. So we've been working with a collaboration called Secure DNA with colleagues from all over the world, mostly cryptographers, on a system that would allow us to screen all DNA synthesis, including desktop synthesizers, without disclosing what it is that we're screening for. And so in the case of the community movement, your biosafety board says you have the right to work on X, that's part of your whitelist. When your lab orders something, anything on the whitelist, even if it's in the hazards database, it still goes through because you have permission to work with that. And the same is true for every academic lab and company. But if you don't have permission, then you won't get the DNA. And in order to get it, you would need to get permission and justify why you're working with that thing, which may or may not be hazardous because there would be some decoys in there as well, and red herrings. So that's one example of how to do it. We can also, even though I strongly advise against thinking about how to do these kinds of things, thinking about large-scale hazards, I really don't think it's helpful. We don't want to train people to have a security mindset and figure out how to weaponize biology. Just don't think that in that direction. It's not helpful. But what we can do is talk about it at the meta level. Maybe it's possible Maybe that there are some things that are really hard for us to defend against, but are plausibly easy to make. And maybe we should not go there yet. Maybe someday it will be safe for us to learn how to do that once we do have better defenses. So this is called differential technology development. And if you're worried about whether it's hazardous to say something, just go one meta level higher.
Okay, that's great, Kevin. And let's just do one more question and then we'll shift into a break. Samir, I see you've got your hand raised. Can we unmute Samir to ask a question? Samir, are you able to speak?
Hi, can you hear me?
Yeah, go for it.
Thank you, Kevin. That was a fantastic presentation. So you touched on, okay, so I think my question is when we're trying to create solutions for these hazards, I think there's, and maybe add one or correct me if you disagree, but there's kind of two paradigms that we could look at this from. Either regulating through an institutional way, like an institutional scale, or personal regulation of sort of creating a culture where people, like you just said, don't really think about or want to do these things. And I think you've sort of touched on both paradigms in your talk and in your response to some of the questions, but would you mind maybe elaborating on the pros and cons of each paradigm? Where are the limits? Do we need both? And is there another way to look at this that's more effective or constructive?
Oh, that's a very large question. I've talked to quite a lot already. I think we need both. I think the hazards evident in the current pandemic and the fact that the current pandemic is pretty minor compared to historical pandemics suggests that we really need all of the above in this case. Downsides of the centralized approach is that, well, bluntly, governments haven't done such a fantastic job of restricting information on weapons of mass destruction historically. So there's been another question in the chat, how do you know, who gets to decide what's dangerous? And the problem is, if you know some way of killing lots of people with biology, we don't want you to have to justify that to lots of people or tell governments about it. So you need some body of people whose job it would be to do this. And ideally, if a handful of people who think about this a lot agree that something is a hazard, then they would be able to put it in the database. Because remember, that doesn't prevent anyone from getting it. It just requires you to have explicit permission from your oversight body. So so it's hard to abuse because it's easy to get around. And that then moves it to the organizational level of permissions. But overall, people still need to notice that they're about to step in it, right? This DNA synthesis screening system is not going to be enough on its own, as people have noticed. Many of you probably have your own DNA synthesizers, and phosphoramidites are probably going to be obsolete in a few years and become extremely expensive as everything shifts to enzymatic synthesis. And at that point, our hope is that we could have screening built into the next gen ones, but there's still going to be old ones. You can still get around it. You can still break the hardware lock on a new gen synthesizer. No single approach is perfect. So I think we need a better culture and we need actual technical measures.
Okay, Kevin, thank you so much for your answers. And everybody, unfortunately, we're over time. So what I would love for you all to do then is if we can get Kevin onto the Slack, we will hopefully be able to engage Kevin more asynchronously there. And then also, you know, again, I'll put Kevin's email into the chat as well, but feel free to reach out to Kevin directly and you can continue the conversation. But although, as Kevin alluded to earlier, certain things don't mention via email is better.